So before we get started, a couple of quick announcements. First, and probably most important, if you are here on Monday morning at this time, I won't be, because it's a holiday, Martin Luther King Day. So there will be no lecture on Monday. I won't be posting anything on YouTube. Speaking of postings on YouTube, for reasons that are still completely beyond me, the recorded lecture did not happen. And so it's not posted on YouTube. My apologies. I did link to the previous year's notes, so you can go and look at those on YouTube. The subject matter is extremely similar. Not identical, but extremely similar. Yeah, your question. Do I know if campus will be closed on Monday? It is my understanding that campus will be closed on Monday. So there may be some services which are available, but as far as I know, campus will be closed. Probably check the PDX website, and it will give you more information about that. Other announcements, haven't gotten clickers from last time uploaded. Most of you have registered your clickers or have told me about them, so that's great. Let's see, the other thing, threw this up here, the Oregon National Primate Research Center, ONPRC out in, well, kind of between Beaverton and Hillsborough, does a really great series of public talks, and these are a few of them. CRISPR primer, they're going to talk about first, and these are Science Cafe, which I think alcohol is available, I'm not absolutely certain, but very much for the general public, but also if you're more interested in CRISPR's general genetic remodeling, etc., gene therapies, etc. And it's going to be at Willow Creek, and here's all the information over here on the side. I think it's a great program. I've been to a couple of them. They're usually really, really good. So with that, we'll move through today. Those of you who printed out the notes may have noticed that I forgot to change this first slide. That happens on a not irregular basis. Yeah, it really is lecture 6, and it really is the 18th. So last time we started talking about DNA chemistry, DNA structure. First, where it came from, nukleinsäure. Yes, you can still buy salmon sperm DNA from scientific suppliers. What are the building blocks? Adenine, thymine, cytosine, and guanine. We'll look more at those a little bit more later today. Chargaff's quote-unquote rule that it's almost always 1-to-1, but at least close to 1-to-1. And then the transformation principle, the transforming principle, I should say, which was degraded with DNase but wasn't degraded with proteases and or any kind of RNases. So certainly looked like DNA being the smoking gun, and then Martha Chase did the experiments, and Al Hershey helped her interpret them in terms of the Hershey-Chase experiment, and again in '52. So do we have any questions about this material so far? We'll talk about Franklin, Crick, and this other guy, and Wilkins, and the whole double helix business. After we clicker questions, exactly. So number 1 for today. If Chase and Hershey had found that S35 was passed from old virus to new virus in their experiment, they would have concluded that DNA was a genetic material, RNA was a genetic material, protein was a genetic material. They had a contamination, the viruses contained protein. And again, if it's less than 80%, I'll let you go again. So if you really want to go again, make sure you pick something you know is wrong. Okay, we're not quite there yet, so tell your neighbors what you chose and why. not going to run into the table when I try and walk that way. Ready to go again? Yes? Okay. We'll all get 100% this time. We're still working 100%. So let's go. And again, feel free to discuss now, too. And do I know why it's counting up instead of counting down now? No. We'll go to a minute, so give people plenty of time. Okay, we're getting much closer to 100%. Why? What does S35 label? What parts of proteins? Cysteines and methionines, exactly. How many sulfur atoms do you see in this model? 0, exactly. How many phosphates do you see in here? A lot. So as I've mentioned before, there are a whole bunch of problems with this model. I know of at least 4, and really great review to come up and figure out what they are. I'm not going to tell you what they are. So yes, C is the correct answer. Why do I always C? Next time when you get the exam, which is coming up soon, just go all the way down to mark C's and everything. I will try to make sure that's not the case. But if in doubt, don't take any points away for guessing. So let's talk about the great, wonderful stuff that Francis Crick did, and Jim Watson happened to be there at the same time. These are the 2 of them, Crick on this side, Watson on this one, in Cambridge when they came up with their model for the structure of DNA. They did 0 experiments. They just looked at other people's data and put it together and made a model. There were some, to be perfectly honest, really amazing insights that they made and came out from that. Start with what did they know first before they actually came up with their model. First one, nucleotide structures. We talked about this a little bit before. We'll go over the nucleotide structure again in just a second. They knew that the linkages between the nucleotides were from 5' to 3'. Molecular biology polarity, yet again. We'll take yet another look at that, again, in much more detail. It has to do with where the sugars are in terms of your nucleotide, and then how each of those nucleotides are hooked together. That was known since the 1920s, but the rest of it, a lot later. Chargaff's rule, in fact, Chargaff had come to visit Cambridge soon before they came up with their model. And rumor has it, whether it's true or not, a lot of the rumors are, where did I put it, in this autobiography that I was going to get the quotes from Crick about. He hated it, but that's another story. That they were really pretty inspired by hearing what Chargaff was talking about. And as theoreticians, they actually had no problem saying, that 1.01 was the same as 0.99. Whereas Chargaff actually did have a problem with that. That was Chargaff's issue. It's not exactly the same. So the other thing, of course, is hopefully everyone knows, they stole data from Rosalind Franklin. And there actually were 2 pieces of data that Rosalind Franklin had at the time. These 2 fiber X-ray diffractions of DNA. And there are a couple of things that they're able to get from this. This was actually the fiber X-ray diffraction of A-form DNA. And this is probably very similar to the data that Linus Pauling had at the same time. And so he actually didn't have as good data, or didn't have access, I should say, to as good data as Watson and Crick did. And I haven't gone into the details, but my understanding is why Wilkins ended up getting the Nobel Prize is because he allowed Watson and Crick to see Franklin's data. But This is the famous picture 51 from Rosalind Franklin. And there are a couple of things that you can get from this if you know what you're looking at. And this is really where Crick's expertise came into this. So Crick was actually going through a lot of the theory, looking at what diffraction of helical structures should look like in X-ray diffraction. So he actually was really attuned to what he was going to be looking at, and again, the whole idea of helical structures. So he knew, basically immediately at looking at this, that this was a helical structure, and that it had a couple of different kinds of repeats. There are repeats at 0.34 nanometers, and actually since this is an inverse space, that's this big blob at the end here, and at 3.4 nanometers. And so these are the structures that they had. They were also, thanks to Linus Pauling, he'd actually shared his data with Watson and Crick's boss, and he had this model for DNA which was totally, completely, and utterly wrong. Which was really, I'm still trying to find an actual image of that one. Apparently it had a triple helix with the phosphates on the inside and the bases sticking out, which in some ways actually makes a lot of sense, but we'll get to that in just a second here. So what do they know? First, nucleotide structure. So the main thing to think about as far as a nucleotide structure is concerned is the ribose. So everything is hooked up through the ribose here. Ribose, 5-membered ring of sugar, starting at the 1', 2', 3', 4', 5'. Remember, everything goes from 5' to 3'. That's where all these guys are linked together. So you have a 5' that's hooked up to a phosphate, 3' OH, and this is how the whole nucleotide is oriented in a molecular biology form. And as hopefully beating the dead horse, the whole nucleotide chain is hooked up together. We also have bases, all of them attached to the 1' position. The other issue, of course, this is DNA, not RNA, so it's deoxyribose, meaning that it just has a hydrogen atom here as opposed to an OH. This is what you have in RNA. Different bases, 4 different bases, A's, T's, C's, and G's, adenine, guanine, which are the purines. The way I remember this, purine, short name, big molecule, pyrimidine, long name, small molecule. So cytosine and thymine in the case of DNA. So what else they have? They had Franklin's data. Again, this is the B form of DNA. They could see that there was a major repeat at this position, and by doing all kinds of fun math, could figure out that that was a 0.34 nanometer repeat, and then there are actually 10 of these, 10 0.34s and 3.4 altogether. And if you add all of these up, each base pair is 0.34, and then one turn is 10 bases. So you have 10 of these, so this repeats every 10. And here, oh no, I'm going to give away one of the problems. If you count exactly 10, it's not exactly lined up with each other. So one of the problems with this model. But in the B-form DNA model, every 10 bases it repeats. And so you're ending up with this structure, which is really nice and symmetrical. So we looked at the nucleotides. Again, how are they hooked up to each other? This is from 5' end to 3' end, 3' phosphate, attaches up to the 5' end to 3', 5' to 3', etc., giving you this sequence of different bases on the inside, GATC. Just a quick shout out in terms of the textbook. This is panel 2.6, which is a really nice way of looking at the individual nucleotides. Another thing that I think I did a terrible job of explaining earlier on, these all start as nucleotide triphosphates. Again, this is the activated intermediate. What I didn't talk about was that these different phosphates have different names. They're called alpha, beta, and gamma, just going away from the sugar. And it's the cleavage here between the alpha and the beta, which gives you your first set of energy, and then the pyrophosphatase between the beta and gamma, which gives you your second burst, which makes this basically an irreversible reaction. So this is really what they knew at the time. What did they come up with? Well, they came up with this model. And literally, it is a model. Again, no data were involved in putting this thing together at all. Wonderful thing about models. Have any of you seen the model in the Smithsonian? It's actually highly, well, when the museums are open, that's a different story, which we won't get into. But the model is actually about this big. It's really pretty huge. It's actually really nice. I highly recommend, if you do have a chance, to go over there and take a look at it. And one of the sections in this book, they talk about, or I should say, Jim Watson talks about waiting for the machine shop to actually make each of these individual pieces so they could finally fit them all together and then line them all up with each other. One thing to notice here, if you look carefully, you should see here a C attached to a G and a T attached to an A. Look at how many hydrogen bonds you have between your G and your C here. Here, the T and the A. Here's also a G and a C, a couple hydrogen bonds right here. How many of you know about this reference? Are you old enough? It's only a model. So that actually is something which we're going to continue with as far as the course goes along. It's a great model, works really well, explains a whole bunch of stuff. It's not what most DNA looks like. Inside the cell, it's all wrapped around and twisted and tweaked, etc. Again, wonderful model, but it's only a model. So what were their big breakthroughs? Couple things. Some things they really could not get from Rosalind Franklin's data or anything that anybody else knew. The first one was that the strands were complementary to each other. So the whole idea of the base pairs, that A pairs with T, G pairs with C. That, brand new. And there was no way to get that from any of the data which they had access to. The other thing, anti-parallel right-handed helices. Anti-parallel, now this is molecular biology for our polarities. So if one strand is 5' to 3', the other one is going to be 5' to 3'. Always opposite polarity. No way that they could get that from any of the data, but playing around with their models was a way that they could actually come up with this. And this is the only way they could get their model, which would then also fit Rosalind Franklin's data. Other thing, as I mentioned before, Pauling's model actually had the phosphates on the inside and the bases on the outside. This makes a lot of sense because if the genetic material and all the information you have is in those different nucleotides, it makes a lot of sense that they're on the outside because that way you can read them, right? So instead, flip them around. And so the phosphates are on the outside and the bases are on the inside. Chemically, this makes so much more sense. If you think about it, phosphates, charged, what are they going to interact with? Water. What are we all bathed in? Water. So makes a whole lot of sense. So phosphates on the outside, bases on the inside. I didn't emphasize this, but these bases are pretty much all ring structures, which have a tendency to be really hydrophobic. So those also really like to stack on top of each other. So chemically and structurally, that also gives you a lot of stability. And then perpendicular in the middle of the helix. This is actually where one of the major issues comes up with the model, because we've never seen that the bases are actually really perpendicular to the phosphate backbone on the other side. A couple other assumptions that they made, which are not as critical. The C4 position of cytosine and the C6 position of adenine are amino groups, and C6 of guanine and C4 of thymine are ketos instead of enols. And the other thing is, just again, to make this structure work, it's not completely symmetric in terms of how the two strands are wrapped around each other. This is just because of the distances that each of these atoms are apart from each other, each of the bonds. And so it's actually offset, which means that you end up with a major groove and a minor groove. And we'll spend a lot of time talking about major and minor grooves, but here's your major groove right here. Here's the minor groove right there. Reminder, their paper is on D2L. I will have at least one question on it on the exam, so read it. A couple other things. Helix handedness. I brought my collection of helices today. Not sure how many of you will still recognize any of these. What did we use this for back in the day? Some of us are old enough to remember. John, I know you remember these. Well, a couple of years ago, I asked people to bring me left-handed helices because everything else we have is right. You know, the DNA is a right-handed helix, the alpha helix is a right-handed helix. So I actually have a whole collection of left-handed helices. And so if you would like to come and take a look at them, please come up and check. If you're curious about right versus left-handed helices, somebody got extra credit for this one because it's actually got left and right-handed helices on it. A lot of fun there. So other point, amino, imino, keto versus enol. So our amino group, NH2, and keto group, O double bond as opposed to an OH. This is the only way that you can get base pairs to happen, is in amino versus keto. These just isomerize relative one to the other. And when we talk about DNA damage, this actually, or DNA repair, this actually will turn out to be really quite important. So again, antiparallel, to me, basically, the two big deals were antiparallel and bases in the middle. That was the real breakthrough, or the two real breakthroughs, I should say, that Crick and Watson came up with. So GC, AT, etc. GC, three base pairs, or sorry, three hydrogen bonds between, again, your keto and your amino group here, and just two between aminos and ketos. Many of you may have noticed if you've looked at old exams, which if you have, this is a really good idea to study for the next upcoming exam. I will ask you questions about base pairs. In the past, dim, dark, distant past, like more than 5 years ago, I asked everybody to draw a base pair on their exam. And then we got 150 students in the class, and it took me about 3 weeks to grade all of your base pairs. So instead, I will give you a question on base pairs. And usually the way that I do it, and you'll see this in old exams, is I give you one that's wrong. And I let you tell me what's wrong with it. So great way to study would be drawing base pairs and memorizing base pairs. Just a thought. OK, questions on Watson and Crick? Yes?
Why is DNA left-handed? What prevents it from being left-handed?
What prevents it from being a left-handed helix? You can try and build it that way. And actually, there are left-handed helices which can form in DNA. I'm not going to get into that. I think Chemistry 492 talks about this. I don't know if it had 492, the biochemistry of nucleic acids. Really cool, wonderful, fun course. But if you look at those left-handed helices, they're way messed up. They've got very different base pairs per turn. They're not perpendicular. They're actually not terribly stable either. So you can make them. And again, this is a wonderful model, but it's not what all DNA looks like. So it's doable, but it's challenging. This is it's more aesthetically pleasing model. Other questions? Comments? Worries? Ready to answer a clicker question? Good. Watson and Crick's original model for B-form DNA had how many? Oops, turn this back on. That would help. Let's go. There we go. Had how many hydrogen bonds between guanine and cytosine? 2, 3, 4, 8, or 10? And I'll give you a hint. It's actually not in their original paper. At least not a diagram thereof. Okay, we have a distinct lack of consensus here, so see if we can come up with consensus. Number A? Okay. Hey, you ready to go again? I've been told what you need to do is just wait until the noise level goes down, and then you can start them again. So go ahead. Make sure again that you vote. Even if you're absolutely certain the first time, make sure that you vote again the second time, because the first time will not count. We seem to be torn between 2 and 3. How many do we have here? Well, you actually in here you can, there's a G and a C right here. 2. So, and this actually, okay, this may sound really nitpicky, etc., but it's trying to emphasize the fact that it was a nice, pretty model. And having 2 versus 3 wasn't pretty enough. And so the whole point here is that it's a model, it's a great place to start, but it really doesn't completely reflect reality, as it were. So the answer is B. Oh, it was B3? Did I mess that up? Okay, the answer is 2. I can change this later, so don't worry about it. I will remember to change it. Those of you who actually did get it correct. So just wanted to go over again a little bit more. Unfortunately, see, this is, well, the nice is I can go back and change the score for everybody. It's hard to change them for individuals. So, but yes, the correct answer, for those of you listening online and didn't click, is 2, not 3, because it's too pretty when you've got 2 versus 3. This model, by the way, actually does have 3, so that's not one of the problems that it has. Another quick reminder in terms of polarity, if you look at the way that the DNA structures, and actually I'm sort of getting ahead of it, the left-handed helix, it's still anti-parallel, even though it's left-handed versus right-handed, because that's still the only way you can get these things to match up with each other. So the whole idea of anti-parallel, here we have, if you just look, the easiest way to see this is where the oxygen is in your ribose. So here, oxygen pointing down, that means 5' end is here, 3' end is here. The base pair that it's base paired with, now the 5' at the top and the 3' is at the bottom. Another way to look at this is here. I prefer this in sort of the space filling model form. Again, we've got a 5' end that's across from a 3' end, 5' to 3', the arrow going down here, 3' to 5' going up here, and then the bases stacked in the middle. I like this structure more than things like this, or even for that matter, Watson and Crick's, because this is that space filling model, the van der Waals interactions. So if you're a protein trying to come up to snuggle up to DNA, you're really seeing something that looks like this, not something that's a wide open structure like this. So it's really still pretty compact, and particularly we have all those bases stacking on top of each other. When we look at DNA replication, we'll see that base stacking is also very, very important as far as that's concerned. And again, now on the outside here, 3' here, 5' there, etc. Another reason to beat on this is when I did have people with their base pair questions, very often when they drew them, that actually drew them parallel as opposed to anti-parallel. Some of you may be wondering what the heck's going on with this A-form and B-form DNA. Why did Pauling get it wrong and These upstarts in England actually get it right, or at least a lot closer to what's going on. It just really depends on actually how much water you have around, whether the DNA is better approximated by the B-form DNA versus the A-form DNA. A-form DNA is also a double right-handed helix. And there are quite a few parts of DNA, actually much more A-form-like. One of my colleagues actually works on a virus where the DNA is packaged inside the virion as A-form DNA. And a couple differences with the A-form DNA. One thing that hopefully should be obvious, actually a little bit wider from one side to the other, but also really compressed, and these bases are nothing like horizontal relative to the other side. It's got a very different major and minor groove relative to each other. Turns out that a lot of double-stranded RNA also will form more in this A form than it forms in B form. And when DNA and RNA form duplexes with each other, they more often form in this A form versus in the B form. So B form, most double-stranded DNA, again, is a good approximation for it. Double-stranded RNA, DNA-RNA hybrids, and some cases of double-stranded DNA, you'll have the A form as well. So what were some of the really nice things to come out of this structure? One of them was that Pauling's idea that, hey, the only way to get access to the genetic information, which is going to be this stretches of ASTs, Cs, and Gs, is if they're sticking out. Well, it turns out you can actually figure out, and when I say you, totally over-anthropomorphizing here, whatever's going to interact with the DNA can actually, just by looking at what's in the major groove, figure out what the bases are in that particular position. And so that's shown here. Major groove and minor groove. The major groove of DNA, depending on what your base pair is, if it's a CG base pair versus a GC base pair, TA versus AT, just by the different hydrogen atoms, hydrogen bond acceptors, hydrogen bond donors, you can actually differentiate just chemically, even when the bases are still paired, in the major groove, what base pair is actually there. On the other hand, if you look in the minor groove, you can't. There's no differentiation here. And the best way of looking at that is going to be over here. Look at AT bases. This would be a hydrogen bond acceptor. There's a hydrogen bond acceptor. Here's a hydrogen bond acceptor. Here's a hydrogen bond acceptor. You can't tell which one is which. So if you're going to have specificity of interaction with double-stranded DNA, it's going to be in the major groove of DNA. And we're going to spend a long time talking about DNA interactions with proteins. It's all going to be happening in the major groove of DNA. Another way of looking at this is what's called the DNA recognition code. No, I don't expect you to remember this. But if you look at the major groove of DNA, and just really totally oversimplifying what we looked at on that last slide, I guess we call it. I actually used to use slides. I'm old enough to have used slides for presentations. Kind of frightening. And you know, there's these glass things that, stuff in the back, projectors. Yeah, well, one of the things floating on Twitter was, what could you mention about your graduate school that students today would not recognize? And one of them was a carousel. So, and if you don't get that, come talk to me afterwards or find John, as the case may be. Sorry to put you on the spot. But yeah, so simplifying this, the idea here is, again, it's all about the major groove of DNA and the different kinds of things that you find there. If I, for instance, were to present you with a piece of DNA or a base pair on an exam, and you wanted to tell me what was in the major groove and minor groove, if it's AT, it's really easy. Why? Because all of you know your base pairs. Not yet. The exam's not for another couple weeks. Thymidine. What's a really different thing about thymidine relative to all the other bases? This yellow thing. Methyl group. Methyl groups. These methyl groups on thymidines are always in the major groove of DNA. So if you're looking at a model like this one and want to find out where the major groove is, find where the methyl groups are. Really easy. And depending on where that methyl group is, if it's next to this side versus that side, that tells you if it's a TA base pair versus an AT base pair. And then where your hydrogen bond donors and acceptors are, just this pattern. And literally this pattern tells you, again, you're coming in from the outside, this foreign protein, sometimes even nucleic acids interacting. And you can form triple helical DNA. There's some actually really cool things about that too. Again, different story. Interactions, the major groove. Minor groove, and sort of the point here is that AT looks like TA and GC looks like CG. So there's no way to tell, just from looking at the minor groove of DNA, what that particular base pair is. Okay, more questions about the DNA structure. Otherwise, I wanted to move on and talk about DNA methods. Okay, not a clicker question. Sorry. There are more later, don't worry. So a couple of concepts, as far as DNA methods are concerned. First one is hybridization. All that hybridization is the fact that you can take two strands of DNA apart and they come back together, and they come back together in exactly this right sequence. So you don't have, pull the two strands apart, they come together in different places. You can get them to do that, but a really amazing specificity of those interactions with the hydrogen bonds. So very, very specific kinds of things. Blots, we talked about Western blots when we talked about protein structures and the protein methods. That's where you have transferred a protein to a membrane, use an antibody to recognize that particular protein. Also use blots for RNA and DNA. Recombinant DNA, this is literally just taking DNA from one organism and mixing it together with another organism. That's one of the really amazing things about DNA. This structure works for DNA from bacteria, from viruses, most viruses anyway. For humans, the DNA is the DNA is the DNA. And so that allows this process of mixing and matching different kinds of DNAs with each other. And we'll talk about how that works. Cloning, not in terms of the somatic cell nuclear cloning, which was Dolly and Polly and all of these organismal cloning. We're going to talk about here, for molecular biology, cloning is just making many, many copies of one specific piece of DNA. And usually it's going to be a gene. And again, my shorthand for that is always YFG, your favorite gene. And we'll talk about how that works as well. One of the ways that people do cloning is you go to, not a big building with books in it, but the basic same idea is you break up all the pieces of DNA in a particular genome and then try and pick out the one that you're looking for. And you almost always are going to use hybridization for this process, picking and choosing which one you want. Libraries are great because usually, and particularly if you're working with a model organism, another reason to work with model organisms, is that libraries are available. So in fact, all of the genes of a particular organism, say humans, and actually you can literally buy libraries that have every single different human gene in them. And they're not ridiculously expensive, just a couple $1,000s. So it's all relative. What do you mean by expensive? So may or may not have a chance to talk about sequencing. If we do, it's great. There are actually a number of different kinds of sequencing, DNA sequencing technologies. DNA sequencing has gotten so fast and so cheap recently. For a long time, the big question is, can we do a $1,000 human genome, 6 billion base pairs for $1,000? Yes, you can totally do that now. People are actually now aiming for a $100 genome. And that is actually what's going to potentially get this into medical providers' offices. So everyone will get you. I will bet again a case of really good beer that if you want, everybody in this room in their lifetime can get their genome sequenced. What do you do with that sequence? Do you share it with your insurance company? There are all kinds of other interesting issues to talk about as far as that's concerned. And yeah, we'll probably not get to PCR today, but we shall see. So first, gel electrophoresis. We talked about this with protein methods. Turns out gel electrophoresis for nucleic acids is way easier because it's got all these wonderful negative charges on it. You don't need to throw in SDS or anything. Every single base pair has got a couple of nice negative charges on it. So really easy to do gel electrophoresis. Again, we'll talk a little bit more about hybridization. Basically, all it's about pulling two strands apart, putting them back together. They come back in the right place. Really, really nice. Blots, again, transferring to membranes and using hybridization to detect your favorite gene. Recombinant DNA, a couple of important things about this. Restriction endonucleases. We haven't really talked about these yet before. Whole idea of a nuclease. Again, it's an ACE, it's a protein, an enzyme. Usually what enzymes do is they chop stuff. What's a nuclease going to do? It's going to chop DNA. The neat thing about restriction endonucleases is they're sequence specific. They bind to and cut an absolutely specific sequence in your particular DNA. And this really allows recombinant DNA to take place because all DNAs are the same. You cut them with, excuse me, a restriction enzyme, you can put them back together. The major thing that, this is great, it's wonderful, but, random DNAs relative to each other is not that useful. Much more useful is the fact that about the same time that people were discovering restriction enzymes, they also discovered these things called plasmids, which are small circular pieces of DNA, which can make lots and lots of copies of these small circular pieces of DNA. Unfortunately, they also carry antibiotic resistance, so a lot of antibiotic resistance bacteria are because of these little plasmids. But it turned out to be absolutely critical in terms of recombinant DNA techniques. And we'll go over some of the libraries, PCR and sequencing, et cetera. Maybe some more bioinformatics too. Why should we care? Well, hopefully we care about our own genome. And again, we're getting to the point that I'm pretty sure that we'll have $100 genomes before too long here. Genetic modification. Right at the very beginning, I showed you the flyer for the ONPRC where they're talking about genetic modification. Some of you may have heard about the reported genetic modification of 2 babies in China. Whether it's true or not, this is the brave new world that we happen to live in. So we should care about that. I would say certainly of all the protein drugs, the antibody drugs, et cetera, which are being made right now, all of these are made through this recombinant DNA technology. So really it's kind of revolutionized drug development. Biotechnology just in general, not just for drugs, research, and go on and on and on and on. A couple things to talk about. DNA. This is important, A, if you're actually working in the lab and working with DNA and trying to purify it, figure out how much you have, but also in terms of thinking about hybridization that we'll look at in just a second. So again, I don't expect you to remember these numbers, but the main thing to think about is if you take a light source over here, shine it through your sample at 260 nanometers, what's going to happen is you will have absorbance of this light that's proportional to the concentration of the nucleic acid that you have in your sample right here, also known as Beer's law, so the molecular absorption coefficient. Really useful rule of thumb is 50 micrograms per mil. If you got 50 micrograms per mil of your DNA, you're usually really happy. And that has a absorbance at 260 nanometers, so in the UV, of 1. That's what happens for double-stranded DNA. Single-stranded DNA, actually, interestingly, has a higher absorption when you have the same concentration. And we could get into all the details about why this works in terms of beta and pi orbitals, et cetera. We're not going to go there. What we care about is that double-stranded DNA has a lower absorption coefficient than single-stranded DNA, which actually is even lower than what you have with a individual base. Now, why do we care about this? Because what we use this for is for looking at DNA coming apart. And so DNA coming apart also is called denaturation. And all that you do here is you have DNA, again, in one of these little cuvettes, like we have right here. And all you do is you have it in your spectrophotometer, like we have in the corner here, and heat it up. And what happens is the A260 stays down, 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 keep heating it up, keep heating it up, keep heating it up, keep heating it up, heating up. And then all of a sudden, and actually this is really pretty sudden, which is one of the amazing things about DNA. Very sudden rise until you get to another area where you keep raising the temperature. It stays at the same optical density, same absorbance. Again, the same concentration here. And what's happening is as you're heating things up, the two strands start to come apart relative to each other. And then there's a specific point halfway up here called the melting temperature, also TM. This case is actually pretty high. It's over 80 degrees Celsius. And this melting temperature actually gives you a lot of information about the DNA. It turns out it's relative to the GC concentration of the DNA that you have. Higher GC, higher melting temperature. Lower GC, lower melting temperature. And you can actually learn a lot about repeated DNA sequences that you have in your genome by just doing this kind of experiment. So a couple of important things again here is that this TM, the melting temperature, will change relative to the percents of GNC that you have. And also this is a very sharp transition from all double-stranded to all single-stranded. So what this means is you heat DNA up, it's double-stranded, but you can also reverse this. If you cool down the double-stranded DNA, the single-stranded DNA, cool down single-stranded DNA, it will become double-stranded DNA. And you can do this back and forth and back and forth. And this is the whole idea of hybridization. And it ends up coming back to the same place in terms of the sequence, as long as you have non-repeated sequences. Repeated sequences, of course, can potentially cause you lots of problems. So, but we'll talk about that when we talk about genome organization much later. So that's hybridization. You're going to need hybridization in the very next step, but I did want to talk about gel electrophoresis. Gel electrophoresis is basically exactly like what we talked about with protein gel electrophoresis. You have an electric field. You put your sample in the electric field, going from your negative electrode to your positive electrode, negative charge because of all the phosphates on your DNA, that matter RNA as well, any of these nucleic acids, and they will move through some kind of gel from top to bottom. The bigger pieces take longer to go through, the shorter pieces go faster. There are multiple different kinds of nucleotide gel electrophoresis. The major ones that are used are either agarose or acrylamide. Agarose is actually made from algae, but it's relatively cheap, although I said to buy some for my lab. It's not that cheap. And makes a nice gel. And it's pretty good for separating pieces of DNA that are between about 500 and 10,000 bases. And that's basically, again, what you can see right here. Acrylamide, on the other hand, is actually also what's used for, you remember SDS PAGE, P-A-G-E, polyacrylamide gel electrophoresis. So same process, makes much, much, much smaller holes. And in fact, if you make these gels properly, you can literally get the difference between a 5 base pair long piece and a 4 base pair long piece, or a 250 base pair long piece and a 249 base pair long piece. And there's some tips and tricks to doing that we're not going to get into here. But on this example here, this acrylamide is, this band right here represents 10 nucleotides long. This one 9, this one 8, this one 7. And in that thing I was telling you about that I was following on Twitter, stuff that students today wouldn't recognize, most biology students these days wouldn't recognize this, even though I spent a lot of time working with these. It's what's called a sequencing gel. Nobody does sequencing gels anymore. We'll look at what that is, and probably at this rate on Wednesday next week. Quick reminder, and I'll try and remind you again at the end of lecture. On Wednesday, I'm actually going to be staying in this room and lecturing for genetics. So we won't have office hours next Wednesday for those of you who are looking for me. But I will be still trying to answer emails, et cetera, over the time there. So last one here, pulsed field gel electrophoresis, PFGE. You'll notice on the side here, these guys are really, really, really big. 2.5 million, 950,000. These are really, really, really big DNAs. And actually, you can use agarose gel electrophoresis to separate chromosomes relative to each other. The only way ever you can get this to work is instead of having a constant field going from negative to positive, you actually reverse the fields with a certain frequency. And what happens is a longer DNA is more confused and has more trouble getting back to a more compact form to go through the gel. And you can literally separate chromosomes, again, using these kinds of techniques. This is great it's wonderful, but we still have one problem, and this is the same problem that we had with the protein gel electrophoresis. Where is the protein? In protein gel electrophoresis, we use stains. We use dye. That's also, again, used for clothes originally to detect particular protein. Where is it? DNA, how do you detect the DNA? Basically, really two methods. My favorite method, again, one of those that current students don't know about, but I know all about, is radioactivity. Radioactivity is great and wonderful, and again, it's what helped Chase and Hershey really put the nail in the coffin for DNA being the genetic material. It's nice because it will expose X-ray film and gives you these kinds of results right here. Nobody likes radioactivity anymore because it's scary, blah, blah, blah. Most people now use fluorescence, and the particular kind of fluorescence is almost always used for detecting DNA in these kinds of gels are intercalating dyes. And intercalation just means it sticks into the DNA, and only when it sticks into the DNA it fluoresces. And so this particular one, the most commonly used, is ethidium bromide, which will stick into and literally between the bases, and then it will fluoresce. Doesn't fluoresce when it's not stuck between the bases. And all you need to do is shine UV light on these DNAs that have ethidium bromide with them, and that will fluoresce. And you can detect that fluorescence just with light. So nice process for doing that. This is great for any DNA, and we'll talk about how you get radioactive labels a little bit later on. But particularly for ethidium bromide, you're going to end up with a whole bunch of bands. Say here, lots of different bands. These are all DNAs of different sizes. And if you're looking at a whole genome, you want to be able to identify which of those bands is the one you want. You might have an idea. I think that I'm looking for the DNA which is, I don't know what this probably would say, this is 3,000 base pairs long. But if you're looking up here, okay, which one of these is 10,000? Is this 10,800, 10,500? How do I pick out the band that I'm specifically looking for? So for this, we use blots. Pardon me. Back up one more. Blotting, again, just like we talked about with Western blots, it's about getting the material. Western blots going to be your protein. Northern blots and southern blots are going to be RNA and DNA respectively. You want to transfer that to a surface, usually the surface of some kind of membrane. Why? The first reason is that if it's inside a gel, it's hard to get to. And the second one is that when you transfer things to a membrane, actually concentrating them. Because a lot of these gels, particularly agarose gel, can be a centimeter thick, half a centimeter thick. Everything's spread out. Whereas if you put it on a membrane, it's all been collapsed into one two-dimensional surface. So how do you do this? You have a gel electrophoresis, separate everything, and then you literally will build a sandwich with your gel and the membrane on top of it. Anytime you're around a molecular biology lab, you can never find paper towels because they've stolen all of them to do their southern and northern blots. So just stacking these paper towels on the top, and what it does is actually pulls through wicking action the solution that you have at the bottom here, pulls it through the gel onto the membrane, and this membrane will specifically stick to your nucleic acids. So you end up just with a copy, just an inverse copy of your gel right here that you've pulled this off of. And this is actually pretty typical. You see this whole dark band right here. If you were to stain something like this is often going to be a genomic preparation of your DNA. With ethidium bromide, it's going to be one big smear. And you actually care about usually one particular gene, one particular sequence which is in there. So you take this, and now you put this in a Ziploc bag very often with a probe of some kind. And here in this case, it's a radioactively labeled probe that is now a very specific DNA sequence. And it will now bind to one of the DNAs in each of these. And then, if you now put this onto some film, you can see a band here, a band here, and a band there. And these are the DNAs which match your specific piece of DNA which you put into the blotting reaction here. So Southern blots are DNA, northern blots are RNA. Why are they called northern and southern? Because southern was the guy who came up, EM southern, with blotting for DNA. All of the other ones came up afterwards. So northerns are called northern blots because it was RNA, not DNA, and western blots were called western blots because they were proteins. I do not know of eastern blots. If you know of anyone, let me know. So these are the ones that we'll look at. And so you can use this wonder of pulling DNA strands apart and putting them back together just by heating them up and cooling them down. You can also use different salt concentrations for this process and get very specific interactions. And the really neat thing is you can actually use a relatively short piece of DNA that will then interact where exactly it matches on sometimes even really, really big DNAs, even as much as chromosomes. And we'll see that when we look at chromosomes a little bit later on. You can do hybridization between DNA and DNA. You can do DNA and RNA. You can do RNA and RNA. Any of these excuse me, work. The only ones which are going to give you a nice stable interactions are the ones which completely base pair with each other. And you can play around with the conditions. Here it's got, formamide, and at 42 degrees you only have perfect matches. 36 degrees you have some imperfect matches. So depending on what you're looking for. If you're looking for a DNA sequence which is similar to the one that you have, then maybe you want to use a condition where you can get a few mismatch base pairs. If you really care about an exact match, you'll use what people will call stringent conditions, where you're only going to get the right kind of interactions with each other. So, got a nice interaction, but how do you know what that interaction is? You need to have some way of seeing what that interaction is. So, in the good old days, back when the dinosaurs were still roaming the earth and I was in graduate school, people used radioactivity for this. Now, almost everyone's using fluorescent labels instead. But the basic idea is exactly the same. You want to have your piece of DNA that you can identify somehow relative to all of the other pieces of DNA. And there are really two ways of doing this. People also call these probes because they're using that to find the specific DNA that they're looking for on a blot. So classic method of doing this is use DNA polymerases with labeled nucleotides. And so this can be a radio-labeled nucleotide. It also could be a fluorescently labeled nucleotide. You pull the two strands apart. You do DNA polymerization. And that DNA polymerization, the red line here, is going to be your newly labeled DNA, be it radioactively labeled DNA, again, could be fluorescently labeled DNA. How that works, we'll talk about a little bit more when we talk about DNA replication later on. If you have this specific piece of DNA that you want to make a copy of, if you don't know what that sequence is, but you have that particular piece of DNA, then often what you'll use is what's called a hexamer primer. And then we also call these random hexamers. Random hexamer is just a mixture of all four bases in random order. So you can have AAAA, ATATAT, TAA, TAA, etc. All kinds of different mixtures in there. What that means is that at least one of those will bind to your DNA through hybridization, and that will serve as a place that you can put in a polymerase and make more of that particular DNA with a particular label on it. Another way that you can do this is by what's called end labeling. If you think about, again, all of our DNAs, they've got 5' ends, they've got 3' ends. There are enzymes that can take the gamma phosphate from ATP, remember, gamma, beta, alpha. This gamma phosphate, which you can literally order from a number of different companies, so they have a radioactive label on it, or it'll have some kind of fluorescent tag on it, and that can be put onto the end of your DNA. How do you do that? Through kinases. We already talked about kinases, protein kinases that put phosphates onto DNA. Polynucleotide kinases will put phosphates onto nucleotides, and polynucleotide kinase does this at 5' ends. Terminal transferase will do this at 3' ends. You end up with double-stranded DNA, now that's got 2 labels on it, one at each end. Usually you only want one of these pieces, so you've got to cut it and then separate the piece that you want. This now is a probe. You can use these for all of your blotting purposes, etc. Later on. Again, nobody likes these radioactive materials anymore. I actually love them because you can follow them around and actually detect them quite nicely with Geiger counters. But there are lots more regulations that go with radioactivity. So what is done now, much more commonly, is basically take your normal nucleotide. So here we have our ribose, our triphosphate, a base, and then some extra piece that's hanging off of it right here. And these can be used by DNA polymerases. They can incorporate these pieces, and then these can be detected instead of by putting them on film using an antibody to this digoxygenin, which is a plant metabolite, or biotin that interacts with a protein called avidin. Happy on blots? Happy on hybridization? Happy enough to answer a clicker question at the end of class? Good. Let's do it when we get there. OK. Blot to detect a specific DNA binding protein. You would call which of the following? A northern blot, a southern blot, a western blot, a northwestern blot, or a southwestern blot? And go ahead and I'll give you 2 minutes. Just go ahead and talk about it. Yeah, and if you're done and you're happy, have a good weekend. I'll see you on Wednesday. If you're not done and not happy, you can stay till 10:05. Five. The suspense. The suspense is, so we're aiming for 100%, right? The answer is E. It's a DNA and a protein. South and western. On that wonderful note, have a great weekend.